Okay, let's dive into molecular identification of species, starting with a technique called DNA barcoding. So I'm showing an image here of fish eggs. So that's what's on the right. Each of these little circles is a different egg. And what I want you to think about are which fish species did these eggs come from, and whether all eggs are the same species or different species. What do you think, just based on looking at this picture? It's hard to say, right? If you asked me, I would say, I don't know. And actually, not just us, but also people who are considered experts in the field would also say they don't know. When I was an undergrad, one of the projects I worked on was sorting fish eggs and doing molecular identification of fish eggs, and you get a vial of fish eggs, like what's shown in this picture here, and they ask you to sort them into vials based on species. There are some species, like sardine and anchovy, those are the only two you can tell just by looking at the egg what kind it is, but everything else goes in a vial they call OFE, or quote-unquote other fish eggs. So that's very unspecific and doesn't tell us anything about what actual species it is just from looking at it. We can't tell from looking at these eggs what type of species it is. And if we do try to guess what kind of species those are just from looking at the eggs, it's possible for us to be wrong and make misidentifications, which can have really negative consequences. So these are 3 pictures of flatfish that are found in California: the Pacific sand dab, diamond turbot, and the California halibut. And when you're looking at adults, the fish, you can tell, right? They look slightly different. You're able to identify what the species is. But when they're at the egg stage, those are fish eggs, obviously. When you're looking at the egg stage, they all look alike, and you can't tell just by looking at them what species they are. So this is actually one of the first projects I did as an undergrad. They gave us some vials of fish eggs. They said, here's a vial of some halibut eggs. Do some molecular work, make sure just to confirm that they're halibut eggs. So I used this technique called DNA barcoding, which is what we're talking about in this video, and I'll explain later. And when I did that molecular identification, it turns out 6 of the 7 eggs in that vial were actually sand dabs. So even these people who were experts were wrong. And the reason why that's important is because they use these surveys of fish eggs to estimate how many adults are in the population, and they use that information to do stock assessments and estimate, for instance, how much fishing they should allow. So if they think all of these eggs are halibut eggs, they'll think there are a lot of halibut adults, and they'll say, oh, you can fish it, it's okay. But really, if they're wrong and the eggs are this other species, we might be overestimating how many halibut there are, and that can have negative consequences for that species. So this is one example of how molecular species identification can help us be more accurate and to provide more accurate assessments of the population. So as I said, when I was working on this project as an undergrad, I used this technique of DNA barcoding. And what DNA barcoding does is it molecularly identifies species that are morphologically indistinguishable. So just by looking at them, you can't tell what species is what. So in this picture here, it's called DNA barcoding because just like a barcode you might scan at the grocery store, a DNA barcode is this short genetic sequence that's from a standardized part of the genome. And so this would be considered our genetic barcode, which is just a sequence of nucleotides, and we can then use this genetic barcode that we obtain from an unknown sample to figure out what species that actually is. And the genetic barcode that everyone uses for barcoding is standardized. So that means if you're doing a barcoding study, you can't just decide you want to use a different marker. The reason why all of this works is that everybody uses the same genetic barcode for DNA barcoding, and that marker that for the most part everyone uses, at least for animals, is a region of the mitochondrial DNA called cytochrome oxidase subunit 1, or that's most often abbreviated as CO1. And here's an image here showing we have our whole cell, we have lots of mitochondria Within each of those mitochondria, we have our mitochondrial genome, and then here specifically is our CO1 gene. And usually about 700 base pairs of that are used as our standard genetic barcode. And CO1 in particular was chosen as the barcode for a couple different reasons. One of them is that, as we've talked about before, some markers have universal primers that are available that work for a wide range of species. CO1 is actually one of those genes that has universal primers available. So what that means is you can use these primers to amplify this CO1 region for multiple different species, which is good because oftentimes when you're getting your sample for the fish eggs, for instance, you might know it's generally a type of fish, but you don't know what type of fish yet. So if you have universal primers that will work to amplify all fish species, that will help you obtain the barcode for those samples. A second reason for why we chose CO1 to use as a DNA barcode is that it has a rapid rate of evolution. And what that means is there are going to be differences in that CO1 sequence between species that will allow you to differentiate species 1 from species 2. If all of the CO1 sequences across all the different fish species, for instance, were exactly the same, the genetic barcode wouldn't do us any good. It wouldn't be able to differentiate which species was which. So you need to have some differences among the species in this CO1 genetic barcode, and CO1 does indeed have those differences because of its rapid rate of evolution. And the other thing that we've talked about, related to what we've talked about previously, is mitochondria are very numerous in the cells. So that means mitochondrial DNA is present in high amounts compared to nuclear DNA. And so since we're looking at a CO1 marker from the mitochondria, it's relatively easy to get enough DNA to use this marker for our DNA barcode because there's so much mitochondrial DNA in there. So how does this barcoding work? What you need to do first is extract DNA from your sample and use PCR primers to amplify that barcode region. So if we were working with these fish eggs, what we would do is isolate DNA from each of those fish eggs and then use universal primers. So like I said, we know these are fish eggs and there are universal primers available to amplify this CO1 region of all fish species. So we would then use those PCR primers to amplify CO1 from each of our DNA samples from each individual egg. After obtaining those CO1 PCR products, what we would do is use Sanger sequencing, which we talked about before. Remember, Sanger sequencing is what gives you these chromatograms, and then we have different nucleotides, so each one's an A. So that Sanger sequencing gives us this sequence, so this would be that genetic barcode from that particular egg, for instance. And then what you do is compare your DNA sequence to a database of known sequences. So say this was the barcode we got from this particular egg, ACACT. What we would do then is compare this sequence from our unknown egg to a database where, so we would have for each different species. So albacore tuna would have this particular sequence. Escolar would have this particular sequence. And so what we would do is compare our unknown sequence to that database, figure out which one was the closest match. Whichever one was the closest match, we would assume that the species was. So if this matched a sandab, for instance, in our database, we would then assume that this egg was a sandab egg. And DNA barcoding is really useful for identifying species in a couple of different scenarios. It's really useful for identifying cryptic species, for instance. So these are actually, believe it or not, two different species of neon damselfish in Micronesia. They basically have no morphological differences whatsoever. Looking at them, I cannot see any differences. And so by looking at them, you can't tell a difference, but if you use DNA barcoding, amplify that region of CO1, compare that sequence you get to a database, you'd be able to match and figure out which species each of these actually is. DNA barcoding is also really useful for identifying species that, for one reason or another, don't have any physiological characteristics anymore to identify them. So that might be the case if you're working with damaged specimens, fish eggs, like we talked about previously. All eggs are, like 70% of fish eggs are roughly the same size, so you can't tell them apart that way. But if you use DNA barcoding, you can differentiate them. And like we talked about before, if you're looking at fish fillets or something like that, where it's some part of the whole organism, so that you don't have the characteristics of the organism itself anymore, DNA barcoding is one technique you can use to actually figure out what type of species it is using that molecular CO1 genetic barcode.